The overall goal of this procedure is to measure a topology of cell adhesion on a substrate with nanometer precision. This is accomplished by first cultivating cells on transparent glass slides and incubating them with a fluorescent marker. Then, the angular position of the microscope's adjustable mirror is calibrated, and the position is stored in the mirror positioning program. The next step is to record the fluorescence images upon irradiation at about 10 different angles. The final step is to calculate cell-substrate distances with a precision of a few nanometers. Ultimately, results of topology of cell adhesion on substrate are used to show differences of adhesion between various cell lines, for example, cancer cells and less malignant cells.
The main advantage of this technique over existing methods like confocal microscopy is that actual resolution is in the range of only a few nanometers. Though this method can provide insight into cell membranes, it can also be applied to other systems such as small vesicles or nanomaterials. Demonstrating the experiment will be our postdoc Petra who will prepare the cell samples, and our engineer Michael Wagner, who will run the experiment.
To prepare cells for variable angle total internal reflection fluorescence microscopy, seed individual cells at a density of 100 cells per square millimeter on a glass slide in culture medium supplemented with 10% fetal calf serum and antibiotics.